Well, welcome everyone, and thank you for joining us for this Arthur Rylah Institute seminar that we've titled Cryptic Calls and Fishy Intruders. I'm James Todd, Chief Biodiversity Officer and ARI Director, and I'll be chairing this seminar with technical support from Andy. I'd like to begin by acknowledging the traditional owners of the land that I'm joining you from, the land of the Wurundjeri people, and pay my respects to their elders past and present. I'd also like to extend that acknowledgement and respect to the traditional owners of the lands where you're all dialing in from today, and to any traditional owners and Aboriginal people who might be joining us. Today's seminars provide examples of creative research approaches to better understand species and inform management decisions, so I'm really looking forward to hearing about that. We'll hear from ARI's Harriet Kulich sharing her collaborative research in the use of acoustic recording tools for monitoring the Australasian Bittern in an environmental water context. And then we'll also hear from ARI's Dr. Ben Fanson, who has worked on quantifying the impacts of carp in Australia and the value of this research in considering management options going forward. We will have time for questions at the end of each presentation, so please, if you've got some, put them into the chat and we'll try and get to as many of these as possible after the presentations. So without further ado, I'll hand over to Harriet. Harriet's a scientist at ARI, or within ARI's riparian and wetland ecology team. Her area of research is as an ecologist focusing on birds and vegetation, and Harriet's current research seeks to inform management decisions by investigating how birds and vegetation respond to environmental water flows in rivers and wetlands, and I'm sure you will enjoy what she's got to present today. Over to you, Harriet. Thanks.
Awesome. Thanks, James. Good afternoon, everyone. My name's Harriet Kulich. My work at ARI, as James heard, is largely around looking at responses of birds and vegetation to environmental watering. Today, I'm going to talk about how we're using acoustic monitoring and some really cool new tools that we've developed here at ARI to improve on our waterbird surveys, as well as looking at ways to get maximum value out of your old data. I'd also just like to acknowledge the traditional owners of the lands where we are today. For me, it's the Wurundjeri Woiwurrung, and also where the fieldwork was conducted, which is across a fair bit of Victoria, namely the Millua Mallee and the Yorta Yorta. So I know I've billed this as talking about new technology and cutting edge acoustic monitoring, but I'm going to actually start off with a little bit of history of birds and water in Victoria. So here in Victoria, historically, water needs, agriculture, and a lack of understanding of the ecosystem have really altered a lot of our native waterways, with many areas kept drier or wetter for longer, shorter, or just at different times than they would have normally. And the flow on of this, if you'll pardon the pun, is that there's been a lot of change in vegetation. For example, we've had death of big eucalypts from being drowned. We've had different types of plants taking over where they were. And we've got issues sometimes with overabundance or the too much growth of dense and reedy plants like common reed or tangled lignum from having lots of persistent standing water. The takeaway from that is that there are lots of different habitat types, or what we call structural habitats for birds in Victoria, but that's not necessarily how it's meant to be. Some examples of some structural habitats are open water, like where these swans and ducks are on the top left, trees like big eucalypts, or in some cases dead trees, like where these little black cormorants are in the middle, and also emergent vegetation, which is plants like grasses and reeds that grow up above the waterline, and they provide good shelter for birds like bitterns and crakes to feed in. So more recently, you might have heard of water for the environment, or environmental water, or e-water, and these are all interchangeable names for essentially water that's delivered by government to hopefully improve or maintain the health of rivers and wetlands, and try to reverse some of those negative impacts that I mentioned on the previous slide. For example, this water might trigger growth of native plants. It might stimulate fish to breed. It could increase connectivity, so allowing fish or plant seeds and propagules to move between pools that are otherwise isolated. And it can also be used to promote the growth of different types of plants to provide the right sorts of habitats for waterbirds to breed and to live in. Essentially, water is pumped or released into historic floodplain and wetland areas, and by adjusting the flooding regularity, the depths reached, and the time of year in which the water is released, managers are able to slowly influence the vegetation and thus the habitats that are available in the wetland. However, for managers to know what sorts of habitats they want to promote, we need to understand more about the birds. So I and my team do a lot of bird surveys in swamps and wetlands around northern Victoria. And this is to improve our understanding of the distribution and the habitat use of these birds. For example, by understanding where different birds are and where they breed, we can look at the sorts of habitats that they like, and thus we can ideally adapt water management to promote the sorts of habitats that priority species need. Now, I say priority species as water management typically will focus on threatened species, as they're the ones which need the most help and support to continue and hopefully increase numbers. In Victoria, we've got about 80 waterbird species, but unfortunately, 23 of those are considered threatened at the state level and 7 considered listed as threatened at the national level. So to do these waterbird surveys, we typically have a couple of observers that go out, walk the area using scopes and binoculars to look for birds, while also listening out for calls. Of course, we're not just looking for threatened birds in these surveys. We want to get an idea of the diversity or species richness and numbers of all species that are using a wetland. So many birds are quite easy to find through these sorts of methods, like a big old swan or a nice big flock of ducks. And this gives us some really useful results for evaluating water management. However, these in-person surveys can struggle to gather reliable information on what we know as cryptic species. So cryptic birds are essentially difficult to find. This is typically because they like to hang out among long reeds and grasses, and they'll hide from perceived threats, such as a friendly ornithologist doing a survey. So these species include a number of wetland birds, such as the Balin's crake, the buff-banded rail, and threatened species like the Lewin's rail and the Australasian bittern. And because of this, they are some of the most difficult birds to survey for, and also to photograph, as you can tell from my personal attempt at crake photography, which the PowerPoint alt text generator has kindly named Close-up of a bush. But with these species, really, you either get lucky and you see one, typically from far away, or maybe you hear one, or you might flush it out by getting a bit too close for them, for comfort for them, like I have with a few Australasian bitterns. And here I can redeem myself with a slightly better photo of a cryptic bird. We also have issues with these in-person surveys with birds that are typically only present in small numbers and that you could easily miss, or which might only move to a wetland sporadically or seasonally. And so they might just not be there when you do your survey, but they still use the wetland. So this poses a bit of a challenge for management. As I said before, to best manage wetlands with environmental watering, we want to understand as much about these species as possible: where they are, when they move, the habitats they need, how they respond to water regimes. So the question was, how could we improve detection of these species so that we could give more valuable information to water managers? Well, another common method of bird surveying is to put out audio recorders at your target sites. The thinking being, if you have your recorders up for a while, any species that you missed during your in-person survey, whether it be because they weren't there at the time or you just didn't see them on the day, they'll move around the wetland and they'll call near your recorder at some point. Recorders also mean that you can capture birds that are more active or vocal at night without having to send people out into the darkness. The recorders let you cover a much longer time period during the year and also over day and night for a much cheaper cost than in-person surveys. And there are lots of studies such as Wimmer et al. 2013 and a meta-analysis by Darris et al. 2017 that demonstrate that adding audio recorders in as well does help to find species that were missed during in-person surveys. However, amazing as it is that we can get all this data and information from such a broad time in the field, once you get your SD cards back to the office, you still need to go through it all. Traditionally, you can either listen through recordings for calls, write down the species you get, or you can look through the spectrogram for matches to the species that you're after. Now, a spectrogram is essentially the visual representation of a sound. You might be more likely to be familiar with the waveform type presentation, which is this grey and blue band sort of in the middle of the screen. So that essentially just shows volume changes over time, whereas the spectrogram, which is the purple and yellow one below it, it does the volume, but it also shows frequency. So you get a lot more info. And if you know what the call that you're looking for looks like in a spectrogram, you can basically just scan through spectrograms looking for that call. So using these methods to find calls is fine if you've only got a small amount of recordings, but we've got big goals. We want to cover lots of sites over a really long time. And so using these methods would mean me spending a very long time sitting at my desk, getting a bit sad and bored because I don't have any time to do anything else. So we've been working with Peter Griffin and Lachlan Francis, who are both my colleagues here at ARI, to develop a computer model that will automate this process, essentially detecting the calls for us and meaning I'm not stuck at the desk going through them all. Now, I'm going to run you through an example of the first waterbird species that we used it for, which is the Australasian bittern. Now, this bird is threatened at the global, national and state level, so definitely a species we want to be picking up in surveys. Now, as a cryptic bird, they're hard to find in our standard surveys, as I was saying before, but they do vocalize, which is great. However, typically only at dawn and dusk and at night, which, as I said, less great for a typical survey. But it does make them a prime candidate for this new automated call recognition system. I'll just start off by showing an example bittern call. This one's from John Graff, which I took off Xenocanto, which is an online audio depository. It's typically a series of low booms. I'll play a recording. There are a few frogs in this, but you're listening out for the sort of foghorn kind of sound. So hopefully you got the gist with that one. And so that one's quite nice and clear, but our field recordings will often be a lot fainter. I'll play a bit of one of our recordings, see if you can make it out. Now I've boosted that one a bit to try and make it easier to hear, but to be honest, still very difficult to hear for me. And if we look at the spectrogram, there's also, you can see one of those individual booms is here, the little yellow bar at the bottom. So to be honest, quite easy to miss those calls, whether you were listening to it or looking in a spectrogram. So the first thing we did was to use Peter Griffin's ARISA program. So that consists of deep learning convolutional neural network models. And I think the name of that alone is a lot to take in for a lot of us. But put very simply, you provide ARISA with an audio file and it'll go through and essentially recognize bird calls using features such as the spectrogram, but also some more complicated analyses. And it'll give you an Excel file that gives you the times it picked up a call, what it thinks the call is, so long as you've trained it first. Now, Peter's used ARISA for a few other species like frogs and bats. So all we had to do was train a new model with example reference calls for the Australasian bittern. So we used a mix of our own recordings and some from Xenocanto that I mentioned before. Now, I'm sure Peter could give you a much longer, hours-long explanation of the specifics of the program, but I'm going to just leave it at that for now and get onto our project. So we got this model trained up, ready to go, but my team didn't actually have any audio recorders back then, so we didn't have anything to do with it just then. But a lot of other staff at ARI do work in those same areas of Victoria. For example, Louise Durkin, who had lots of recorders out for frogs along the Murray, which I've marked in red on this map. Now, these were great for Lou for improving our understanding of frogs, but there's no need to limit what we use our recordings for. So she very kindly let us pinch them, and now we've been able to reuse them to look for evidence of bitterns. So we actually got a fair few positive detections from those recordings, which is really exciting. But we can't just go off and use that info without doing a bit of checking first. And this is where Lachlan Francis's tool Ariel comes in. So Ariel's a really handy user-friendly interface for validating or checking that the calls that ERISA detected are correct. Again, I'm just going to give a very brief explanation here, but the ERISA program from earlier spits out those results, the Excel file marking the time of each call, which file it's in. And so Ariel will read that Excel file and the source audio files, and it gives us this screen. So here we can see a spectrogram of the first recognition in the file. It tells us what it thinks it is. We can press play down here so we can check and see if we agree. And if not, then we can say what we think it is over on the right. So this screenshot's actually from quite early on in the process. Hopefully it's found your target species, but in this case it was actually frogs. So I've clicked frogs here, and then you just go to the next one. And because we're only doing presence and absence at this stage, it's also got a really handy feature that lets us skip validation for all other recordings at the same site. So once you're happy you've got one positive detection there, you can just skip on to the next one. So as you can imagine, this really speeds things up a lot, but I'll give you some stats on this. We had 818 days of audio recorded to go through. It took about 4 days to create the model, 8 hours to verify, and so all up about 4.5 days to analyze 818 days worth of recording. And Pete and Lockie are getting better and better, so these times are improving. I also want to note as well, obviously you don't have to recreate the model every time we want to look for bitterns again. If I come back in with a bunch more recordings, I can just plug that back into the same model and have it go. So very efficient. So what did we find? Well, looking at our map from earlier, these are all of the sites which had recorders from Lou's project. And these in green, I'll just zoom in as well, are the ones where we got positive detections that we validated as correct for Australasian bitterns. So 10 out of about 130 sites. So this demonstrates proof of concept that we can do acoustic recognition models for bird calls. And it also gives us a really solid baseline index from 2021, which is when these recordings were from, for presence and absence of bitterns across the floodplain surveyed. Now, even though this was more a test than anything, this baseline is actually really useful for long-term monitoring throughout different water regimes, and it's established this as a thorough and a time and cost-effective method for future monitoring. It also gives us some great data that we can add to databases such as the Victorian Biodiversity Atlas, or the VBA, where it's available for use by anyone, including the general public. So our next steps are to build a more comprehensive waterbird model. So we've learnt lots about model building, so what's important, like having good quality recordings and a range of different recordings. And we're going to use this to try and build this more comprehensive model with as many species as possible, particularly focusing on those cryptic waterbirds. For the most part, this will mean finding as much training data or calls as we can so we can build these models. And then we want to use these to complement our in-person surveys to pick up anything that we might have missed. Now, I think it's really worth noting that we don't anticipate acoustic recording replacing our in-person surveys, but rather complementing them. So acoustic surveying is really useful, but it does have limits. For example, it won't detect species that don't vocalize, and it doesn't easily give a number of species, just the presence or absence. It also doesn't really give you the same insight about the birds or the wetland that you can get from being in person. For example, you won't be able to see if there's breeding behaviors like nesting or chicks present, and you won't get a same understanding of the water levels or the habitats that are around. However, in conjunction, we believe that using the waterbird acoustic recognition will be really beneficial to detecting additional species to benefit completeness of our surveys. Now, I might just tell you a bit about what we hope to find. Firstly, it'd be great to get a better idea of the distribution of some of our rare and cryptic species, like the Australian little bittern, which is different to the one I was talking about before. And we've sort of sporadically found them at different wetlands in different years of our surveys. So being able to cover more area and more time should let us get a more solid idea of their range. And range extensions have been made using audio recordings, so it certainly is a viable method. Would also be great to investigate interesting ecological questions, such as looking at how acoustic phonology, which is the timing of calls, and species richness might vary in response to management actions, as well as natural events such as floods or topically avian influenza, which might be coming to Australia soon. So what are our takeaways? We've established acoustic recognition models as a viable method for efficiently surveying for cryptic species, and they can cover more time and area than you could practically with people. We've got this reliable baseline index of presence and absence of Australasian bittern at our surveyed sites. And because we use those frog recordings to do it, we've demonstrated a great way to use existing data and to effectively collect data and answer questions from before we even considered the project, which I think is really cool. And overall, it's given us this great new method to efficiently gather information so that we can better inform water management and better protect threatened birds in our wetlands. Thank you.
Harriet, that's fantastic. I just think this is so exciting, and the combination of bringing the kind of the IT smarts together with the ecological nous is probably the key here. So, it's well done. Thanks for the presentation. Just a reminder, folks, you can put some questions in the chat for Harriet if you so wish. But Harriet, maybe one from me. You mentioned that it's most likely to be of use for, or this method or approach is most likely to be of use for presence absence of species. And I think you made a comment there that it might be a bit limited in terms of giving us some idea about the number of species. But do you think it's got that potential in the future to help with thinking about the numbers of species, particularly as they move around the landscape?
Yeah, no, that's a really good question. At the moment, we're not really able to get definitive numbers, as it's pretty difficult to tell whether different calls are coming from different birds, unless you just get one, I guess, and then you can say one bird. But otherwise, I guess you can use sort of call rate or like the number of calls to get a rough indication of a category. So you could say, lots of birds or not many. But people are definitely working on that, which is going to be really interesting. There's also a really cool method that I haven't tried personally, but I've heard a bit about, which is essentially you set up a few recorders at one location, kind of close to each other, and then if something calls within range of all of them, you can triangulate the position that it called from. And then if you had a few different things calling and you could see that they were, a few hundred meters apart, then you could definitely triangulate a rough number. So that'd be best for, small numbers like the bitterns. I think you'd struggle with, flocks of parrots. But no, that sounds really cool. But yeah, like I was saying, at the moment, we're mostly looking to supplement our in-person surveys or potentially look at range extensions. So for now, yeah, just happy with the presence absence data is going to be really useful for us for now.
Yeah, no doubt there's lots and lots of questions that could be asked, and how far we can push the technology will be really interesting to see over the coming years. Actually, Peter Griffin, who's obviously been a really integral part of this project in terms of the audio recording and the software to identify calls, has put something in the chat there around painted snipe. And so maybe there is actually for people on the call, we know that people have got potentially have recorders out in the landscape. So if you are recording species, getting access to those audio files might be really useful to help, particularly for some of these more cryptic species, trying to identify them in the landscape.
Yeah, I think painted snipe, which Peter's talking about here as well, as species we don't really have, or no one really has, as far as we know, a good recordings of. So I think we're hoping there's a few. They're more up in New South Wales, but they occasionally turn up in Victoria. So yeah, it'd be great to try and get some recordings of those, and then see if we do have more around than we know.
Cool. Andy, you're keeping track of questions in the chat? Can I hand over to you?
Sure can. I think, as you said before, there's lots of questions, but they're not coming through thick and fast just yet. I'm imagining people are still formulating the best way to ask them. I do have a question from Paul, though, and it's about the calls that you detect, whether or not you can learn something about their behavior ecology from the types of calls. So he's asked particular about attracting a mate, or if they're nesting. Do you get any sort of that ecological understanding from the calls?
Yeah, that's a really great point, Paul. I think one of the things I would be interested in looking at in the future is, I think you talk about breeding in particular. So some species do have specific calls they do for breeding. So I think it would be great to look at, are there variations maybe in when that starts? Then you can compare environmentally watered wetlands versus not watered wetlands, or even times of drought versus not drought, or any other sort of factors that you want to consider, really. So I think it's definitely a really, really exciting space, I think, yeah.
It sounds like there's huge opportunities there, Harriet. Question from Brad, though, about things that might trip you up. Do you get cryptic, do you get species that mimic other birds and make it really confusing for the model to determine what species it actually is?
That's a good question. I think what we probably all think of first is lyrebirds. So, and they obviously are big on mimicry. Not typically present in our swamps, fortunately. So not an issue that we're aware of at the moment. There definitely are species that sound quite similar, but I think Peter's found that I think it's sort of, the models can be really good at picking up differences that we don't necessarily tell ourselves. So so long as you can distinguish the calls when we're training the models, so that we give it the right calls and tell it that this is a white-faced heron, this is an egret, it'll learn that, and it'll probably be better than us, to be honest, in some cases. So yeah.
Thank you. One final question before we go to Ben. So actually it's just come through. Is there a place where other researchers and monitoring staff can upload monitoring records captured for their own purposes, but make it available for other people?
That's a very good question. I know there's a lot of spots that, you can put up recordings of a species call, for example, Xenocanto, which I mentioned before, or places like eBird. You can lots of online sort of depositories, but I'm not sure if there's a particularly well-known spot for uploading like a really long duration recording. But by all means, if you have something, feel free to email me, and we can see if there's something, if we can look through, or maybe there is somewhere you can put them as well.
IT is going to be on to you, Harriet. Your inbox is going to be full.
Going to need some more hard drives. Yeah.
Yeah. All right. Thanks so much, everyone, for the questions. I'm going to throw it back to James.
Thanks, Andy, and thanks again, Harriet. That was a fantastic presentation, and as I said, really exciting development in the use of remote or close sensing approaches to monitor waterbirds and a range of other species. Really look forward to seeing where that goes in the future. Our next presenter is Dr. Ben Fanson. Ben is a senior scientist, biometrician at ARI. So his role is really about providing scientific and statistical support for a range of projects across the institute. So he works right across different teams in providing that really important statistical and biometrics input into a range of projects. Today, he's presenting on his recent research on quantifying the impacts of carp in Australia and some of the management implications. Over to you, Ben.
Much thanks, James. Hold on. All right, can you see that, James?
That looks perfect. Thanks, Ben.
Oh, fantastic. Thank you. Much thanks for that introduction. This is fantastic. I love presenting my stuff. I think this is what I enjoy most, is communicating the stuff, the research that we do. As James said, I'm in the quantitative area. I'm a biometrician, so I'm not a fish ecologist. I'll just put that up. That's a caveat. But I do work in a fair amount of the fish area. But there are other people, a part of my team that I'll talk about in a second, that provide that fish ecology background. All right, just quickly, I do want to acknowledge the lands that I'm presenting from, the Wurundjeri people, and I do honor their elders past and present. With any good research, the quality of it is based on the team that you work with, and I had a great team working with the research that I'll be presenting here. So these provide a variety of backgrounds. It's inter-organizational for different organizations, and a variety of research from landscape ecology to fish ecology to management. So the stuff I'm presenting here, a lot of the kudos go to these fellows. Another little housekeeping thing is the research I'll be presenting is based off of basically 2 papers that we have published in biological conservation. The first one was in 2021. This was on common carp, and the goal of this paper was to estimate how many common carp there are in Australia. You know, nice small little project. And then the second paper is thinking about converting those densities to what kind of impact have common carp had on the Australian environment. And that's what I'll be presenting here. All right, so we just had a fantastic talk from Harriet, which was great. I loved it. She was talking about biodiversity and wetlands and thinking about e-water and stuff. For my talk, I'm actually going to go under the surface and look at carp, a threat to biodiversity. And I swear that's the only obnoxious eye-rolling animation you'll get. So it's out of the way, there you go. So as we get going, I need to provide some background info so that you understand why we did this study and how the study has, what kind of impact the study, our results will have. So first I need to just talk about common carp, a natural history of common carp. What are they, and etc. So common carp are native to Eastern Europe and Western Asia. And they're large fish. They are usually between 2 and 14 kilos, but they can get to 30 plus kilos and up to 80 centimeters. So we are talking large fish in the environment. They prefer freshwater habitats, usually slow flowing or static environments. So wetlands, lakes, and rivers, larger rivers that flow slow. They are rather robust in terms of their niche. They can occupy temperature ranges from 2 to 40 degrees Celsius. They can survive in frozen lakes, in the deep sections of the lake, in cold water. They can tolerate salinity up to 0.5%. And on top of that, they can also survive in low oxygen, hypoxic water environments. They have a behavioral, physiological ability to gulp air, and that provides them another way that makes them robust. In terms of their diet, when you look at carp, you can see their mouth, and you can see the large mouth and how it bends forward. They're bottom-feeding animals. They are omnivores. They will feed on aquatic plants, and then they also will be in the mud, digging through the mud, and they'll get to the tubers of the plants, completely destroying the plants. They're also looking for benthic inverts, as well as mollusks and aquatic insects. And they'll actually even eat small fish. And so they do have a very wide diet. All right, so that gives you a little bit of background of common carp. All right, so now I'm going to move into from their natural history to their unnatural history. Common carp have spread throughout the world. They're on every single continent except for Antarctica now. They have spread throughout Europe, over to the US, down to South America, and then obviously over in Australia. As a consequence of their amazing spread, they are considered one of the world's 100 worst pest species. How did they spread? Well, chalk it up to humans. As humans migrated, they brought carp with them. Carp in Europe is actually an angling species, much less here, and they are used for food. So when people migrated, they brought carp with them. They would introduce them to the rivers so that they could feel like home and go fishing. They also would raise carp in farm dams and other fish farming techniques to raise them for food. Also, as I mentioned, they're really robust, and so if you put them in a in your pond in a garden, they'll usually survive, even if the water quality is awful. Once they're in these areas, they've actually spread with humans as well, but also through natural mechanisms, flooding. So even if you have carp in an isolated farm dam, if flood comes through, they'll get out and they'll get into the river system. Also, they found that birds can actually move carp, in particular duck, through the environment. It's actually the eggs that are getting moved. The ducks will feed on the aquatic vegetation, ingest carp eggs, and it'll actually make it through the alimentary canal and through the feces, and they can actually survive. And they have been thought to be wise how carp have gotten into far-flung places. All right, so there's the general. Let's get to Australia. So carp have been introduced since the 1850s, but they never really caught on and really spread. It wasn't until the 1990s, and especially the mid-1990s, there were three main strains. One was Prospect, which was over in Sydney, Yonko, which was over in the Yonko, but the Bulara one is the main strain that really spread through the environment in the 1960s. The Bulara was down in the Gippsland area, but there's also several of these fish moved into farm dams around Mildura. And so what ended up happening is we started to get spread down in the Gippsland over in Mildura area, and then we had that Sydney Prospect strain. And as we went over in time, we kept getting spread. In the 1970s, there was a huge flood up in the Mildura area, and it just brought carp throughout the Murray-Darling Basin. And then we kept spreading and spreading. And so carp have just made it throughout Eastern Australia and a little bit down in Tassie, as well as, I didn't put in this one, over in Western Australia in the 2000s. So our first project was going through, doing that distribution and getting some estimates of density of carp throughout this area. And as you can see, it's bloody scary. There are a lot of carp and at some high densities. And these densities, you can see definitely, especially during flood years, where the numbers are just phenomenal. This was taken by Iva Stewart, one of my collaborators, near Torumbury, and you can just see the mass amount of carp backed up by the weir. And then if you get into the fishway, you can just see all those juvenile carp. Obviously, a lot of these will die, but the impact is got to be large on the environment. And that's where this study started to come in. What is that ecological impact? We have so many carp out there, and they're in the environment, and they can change, and they can have a big impact on the aquatic systems. And since carp has been spread for a long time, there's actually been a lot of studies out there looking at the variety of different categories of carp. I'm just going to focus on a couple here because these are the main ones. There's a negative effect of carp in the environment. They've seen that as the density of carp affects the amount of aquatic plants, they go down, and the amount of macro invertebrates. So positive effects, positive in quotes here, it increases the turbidity of the water as well as the amount of nutrients in the water. It makes the water systems more eutrophic. And when you have eutrophic water, you often will get more zooplankton and phytoplankton. So they're ecosystem engineers. They're changing the environment that they're in, especially in the densities that they're at. And it's not surprising with this many carp out there and these impacts is made the news. The public is well aware of the effects of carp, and you see these mass kills, you'll see them changing their environment. And so there is a push, a public are aware of the impacts that are out there. As a consequence, carp control is of interest. There's different mechanisms of carp control. The first one, physical removal, is the most straightforward. It's also expensive, and there's a lot of limits to it. Physical removal will be through wetland netting or carp bash, annual fishing competition. Iva Stewart's come up with some cool fishway traps that take advantage of carp's jumping behavior to catch them. This has been done and successfully down in Tasmania. In the 1990s, carp were introduced to Lake Sorell and Lake Crescent. They think maybe by anglers using live carp bait. And the numbers popped up in the late 1990s. A whole bunch of effort was put to trapping and catching these fish using the Judas method and whatnot. And they were able to get carp out of the system. Lake Crescent, it was, I think, 2009, that was carp free, and Lake Sorell right around 2020. And so that's fantastic. You got a non-native species out of the system. And of course, the trout community is the one most happy, another non-native species. Anyways, so besides removing them out, there's also physical barriers, ways to prevent the spread, to keep numbers down. That would be through carp screens, or where I'm from the Midwest US, and there's a concern of the Illinois River, which is connected to the Great Lakes, of keeping carp out of the Great Lakes, and they use electric fences over there. They aren't decreasing the amount of carp, but they're preventing the spread. To actually reduce carp, there's other technology that's advancing. There's proposed genetic ones, Trojan Y, which ends up killing male offspring, daughterless, which ends up killing female offspring, and these are driven through gene drive, so all the offspring have the gene. Those techniques do involve genetically modified organisms, and they have their own risks associated with it. Another mechanism, technology, is biocontrol. Obviously, Australia is familiar with the rabbits, and in particular, the carp herpes virus. And with any technique you use, you need to think about the cost benefits. And that's what's happened with the carp herpes virus. It has become a technique that we've been researching, thinking about the cost benefits, a risk analysis of it. And this is through the National Carp Control Program, which started in about 2016. This is a very expansive project, 15 million on it. It had 30 some projects, over 40 plus researchers. The main bit of it was run from 2017 to 2019 to gather the information, and obviously political issue. But there's a lot of research that was done trying to figure out, can we release carp safely? Part of that program was looking at the ecological benefits. If you're going to reduce carp, what does the public expect to see from a reduction in carp? What can we say about what kind of effects we expect to see, in particular, ecologically? They did do a research paper on this as part of the NCCP. What ended up happening with this project is a lot of it was basically based on expert opinion. And so you can see a lot of what was come out, the responses of how a carp reduction would be, it'd be a moderate benefit, or a moderate large benefit, or a large benefit. But it's really hard to work with those rather qualitative assessments. From moderate to moderate large, how big of a change is that? From moderate large to large, how big of a change is that? So it's limiting in its nature. And this was pointed out by other researchers. Koff paper, for instance, basically argued, we just don't know what's going to happen if we do these carp reductions. Will we see the benefits? Because we just don't have the information there. And that's where this study comes in. We're really interested, can we take these responses, these more qualitative, and make them more quantitative? And there is a way to do that if we have the data, and carp actually has the data. And that's the idea of these density impact curves. As the densities go up, what kind of impact does it have? And these are used in other fields and are a fantastic quantitative way to really look at those relationships. And so for here, we can see as density goes up, you start seeing impact. And it's really when you get to the really high density that you see the impacts. And so, for instance, if we take a scenario where we say, cool, you have a system which is at the high density, if we do a 20% reduction, we may expect to see, go down the curve, an 80% improvement. So that would be fantastic. That would mean that when you did the carp reduction, that would have a strong benefit. However, the curve doesn't have to be shaped that way. It could be shaped where all the effects are at the low density part. So you've already moved up the system. And so by the time you get to here, if you do a 20%, you just get barely any improvement. So you shouldn't expect much. And so when it comes back to the National Carp Control Program, they estimated around 40 to 60% would be the carp reduction associated with the virus. There's a lot of caveats here, depending on the system, temperature, water, blah, blah, blah. We're just doing a simplification just to show kind of roughly the ideas that could be used here. So if there was basically a 40 to 60% reduction, you would start at high, go down here. You could either get a really strong improvement or you may get nothing, depending on the shape of those curves. So And it also depends what the density is at each of those locations on whether you see that effect. It may be at mid density, you just don't see any effect because it's only at high. So you really need to put this into context of density and what these curves look like. And so that's what this study was aimed to do, is to go through each of these carp where there was enough data and try to build these curves. To do that, this is a complete desktop study. We used the literature that existed out there. So did a literature search, we'd go and find a paper, look through the paper. Often we would have to grab out a table to figure out what pre and post changes in carp biomass, extract out those relationships, and then we run what was called a meta-analysis. We take those information, those effect sizes, those changes, take the uncertainty with it, and pull them together to try to get a general change. All right, what did we find? So we were able to find 41 studies that fit our criteria of what we needed for the meta-analysis, and that was across 13 countries. And from that, with these 41 studies, often they had multiple impact categories in there. And across those, we were able to identify 8 impact categories with enough data. In the biotic area, macroinverts, macrophytes, phytoplankton, zooplankton, and the abiotic suspended solids, turbidity, nitrogen, and phosphorus. And so for that, with all that, we're able to build those density impact curves. Here we use biomass as a metric for density. And we looked at standard impact. So for turbidity, they all started at 1, that's 100%. And then we could see at 2, I mean, there was a 200% increase. So we see that around 200. For below the curve, we're seeing a decline. So that'd be, if we're down to about 50%, it would be about here. So you see a half of it. So there's a little bit of asymmetry there, but there's a little bit of rhyme and reason to that. So if we go back and think about, let's just say on average, where there's a 50% decrease due to the carp virus that was estimated from then CCP, we could see that for turbidity, you get about a 30% decline in terms of the how turbid the water is. But if you go over and look at macrophytes and macroinverts, you're going from about 1% of what was originally there to about 10%. So you're not getting a huge gain in terms of that. What you would have to do is probably get closer to 90% decrease to get back to a roughly original, or 50% of the original amount of macrophytes or macroinverts that were there. So part of any prediction, you got to have some kind of validation. What we try to do here, we did some of the normal leave it out kind of approaches. We also found studies in which there was carp removal and we made predictions of those studies of what we think the change would be, the response to that carp removal. And what you can see for each of these is that the gray is the prediction, and these data are not in this prediction, and the blue are those carp removal studies. What we can see is, in general, we get a decent overlap. They're usually within 10 or 15% of the prediction. There's a couple that are on the outside. Macrophytes, we did less well on. What we ended up doing was under-predicting how well the macrophyte would respond to that. So they actually were more macrophytes in the end. Cool, so that was our first step. What we thought as a proof of concept, we would actually try to predict, so what kind of change do we think happened to Eastern Australia since the carp invasion? So what we ended up doing was, from our original paper, we took a carp biomass layer. So we have our densities of what we think is through the system. And then we took this paper, those density impact curves, blend them together so that we can make a prediction and look at where we think the highest impacts were through the system, yellow being higher impacts. So let's see that in an example. So we did that for each of the categories. And so we made a lot of assumptions here. It's more of a proof of concept, and there's a bunch of ways to improve it. But it's a start. It's a way to say, cool, let's think about it. Let's try to use this in a way and provide an example. And in our paper, we try to do a really good job of identifying where we think limitations are and what needs to be done next. But for here, you can see with, so this one, SS, is suspended solids. We think our model predicts roughly around a 35 to 40% increase in the suspended solids in the system on average, with bigger systems having more weight and whatnot. And then you can move down the system and go down back to the macrophytes, and we're predicting about a 40% loss of macrophytes in this the waterways and water bodies as a consequence. So for each of these categories, we have a map. So we just produce a map and we have our impact category. So for macrophytes, the biggest impact, yellow, would be loss of macrophytes. So it would be the percentage change. And we get up to some 90% change in some of the areas associated with that. And so for each of those categories, we're able to produce those maps. So where are we heading from here? We're looking for ways to validate the predictions more. Obviously, our validation is just a very initial first step. There's much better. We needs to be pushed and tested so that we can figure out what's wrong with the model, ways to improve it, and whatnot. We're also trying to figure out ways to quantify the higher or similar trophic levels, fish, waterfowl, turtles, and stuff. It gets difficult here because you can't really group them in the same way. Often when there's similar or higher trophic levels, each species kind of does different things, and there's not much data out there. So we're trying to come up with ways to see if we can build some kind of similar curves, maybe trying to combine what we're doing, what we hear with expert opinion to actually try to make curves so that we could fit it into the modeling process. All right, quick recap. So I provided a little background. Carp are a major pest species, and they've invaded Australia. We need management strategies to deal with them. To do that, we need to understand the ecological impacts and the benefit and the potential benefits of that management action. We had identified a knowledge gap. Those quantitative relationships were missing, and we remedied that by going through the literature, creating those relationships, doing it for the categories that we could find data, and then we provided an example of how to use those relationships so that you could make predictions with it. Cool. And that is all. Thank you.
That's great, Ben. Thank you. There's a lot of really great science in that presentation for starting to try to come up with some management strategies for a pretty, quite frankly, almost unmanageable problem. So trying to make headway on that is really useful. And so that's the science that you've been collaborating there on is going to be really important to that. Just a reminder, folks, there is a bit of time for some questions in the chat, but maybe one from me, Ben, before we go to others. The impact of the management on some of those other benefits that you were kind of modelling does it also take into account the effectiveness of that management action in terms of how long the management action, the effect of the management action might last, or is that another piece of work?
That would be a completely another piece of work. So yeah, with our study, a lot of the studies that we had were long enough to see the response. They only publish it after that point. So we don't have after 6 months what it will look like, or after a year what it will look like. So a lot of these are on the longer side associated with it. And that's just the limit of the data that we could get. We just don't have that kind of resolution. It'd be fantastic to give guidance on, be like, cool, if you reduced it for a year, this is what you'd expect. But this is more probably at the end bit of it.
Yeah, understood. No, that's good. So I'm just thinking, there is a range of things that will go into management decisions, including, the longevity of the effectiveness plus cost and things like that. But that's great.
For sure, exactly.
Okay, Andy, some questions in the chat there.
We've got quite a few coming through, so I'll do my best to keep up. The first one, Ben, is about the relationship and sensitivity between carp mortality and ecological benefits. They're wondering if the relationship was likely to be linear or a threshold where sort of things happen very quickly, as in your sort of graphs you showed.
So carp mortality, is that, decrease in carp density, or the actual bodies that exist in the system and that effect?
It's a good question. I'm going to assume it's the reduced in the density, reduction in density.
Yep. I mean, that's exactly what we were trying to do here. So originally, it was often worked with thresholds, just say 250 kilograms per hectare is the threshold. And if you got the population or the density down below than that, we should see a bunch of ecological benefits. What our study here shows, it really depends on what you're looking at. And each of those curves provide that extra information. It's not just a threshold. It's actually a lot of them are fairly linear. But there's a few with the macro inverts and the macro phytes where you really have to get them way down there. So you can really, the advantage of all these curves are you could play with the ones you put the highest weight on and what you think are the most important, and you can decide from there where you think the threshold is, where you needed to get it down to hit your management objectives.
Perfect. Thanks, Ben. This one seemed like just a funny question, but I think there's actually a fair bit behind it. Have you recorded the sounds of carp? I'm just thinking this could be a way of understanding density, kind of things Harriet was saying. Have you looked into anything with acoustics?
No, that's not me. That fish ecologist. I have no idea. I mean, fish make noise. I mean, obviously, there's a whole bunch of area over in the coral reefs and stuff, and looking at noise as a way to get an idea of what fish are there, and also use it to attract fish to new corals reefs. So the acoustic sounds are important. I have no idea for carp.
I can jump in super quickly.
Ooh.
People can't have looked for fish. I don't know that much about it because I don't do fish either, but it is a thing people can do in acoustics, and there are like underwater recorders as well. So
awesome. Thanks, Harriet, and thanks, Ben. This next question, you mentioned how having different frogs and the other species, you had different impacts on them based on their trophic level. This question is actually about plants. Do different types of plants have different responses to the carp impacts?
So the vast majority of studies that I've looked at, so we actually have a lack of studies, I'd say, in Australia. A lot of them are international. There has been a really generalized decrease in the macrophytes across a broad category of species. I'm not going to say that all of them, but there is a very general response to it. And that's actually fairly common with lower trophic levels that we see with these density impact curves. You can usually predict them pretty well. There's a fantastic meta-analysis of, I think, of 4,000 studies out there, and they show really consistent patterns with the lower trophic levels. It's those higher ones that we really see the difference. I'm sure there's outliers. There's always outliers.
The data is always messy. Well done. Question, this has surprised me as well. Do carp eggs really pass through the gut of waterfowl? This is a bit more of an ecological question, but you'd know better than us.
That's what the studies have shown.
Wow.
Yeah, they can actually make it through. So there is some resistance to the through the system, the alimentary canal of the waterfowl. So yeah, I mean, I can't remember if it was PNAS or something. It was a decent paper because everyone was surprised. No one thought it would be done. Yep. Yeah.
Following
cool.
up on that then, do you think what they did with Lake Sorel, where they eradicated them from that lake, might only be temporary if they could be reintroduced by birds visiting that lake again?
I mean, there's always a chance. Exactly. I don't know. Sorry, I'm not as familiar with Tassie in terms of how many carp are in farm dams and stuff. I know they were for a bit. I don't know if they're out. So once you get further distance, the further away the source of the carp is, the less likely that you're actually going to get those to stay in the duck log enough before it's passed through in the feces. So yeah, so the further away the closest carp population is, the less likely you'll get it.
Awesome. All right.
Andy, I reckon we might be out of time. People
perfect.
might have 2 o'clocks to go to. So first of all, thank you again to Harriet and Ben for such interesting science and great presentations, and presenting that in a way that I think everyone could relate to. So thank you very much for the work you put in there. I think there were a couple of questions, maybe Ben for you, perhaps Harriet as well, but for those on the call, feel free to follow up with Harriet and Ben offline directly, and they might be able to have that respond offline. But just a reminder that our next seminar will be on Wednesday, the 20th of November from 1 to 2 p.m. We have 2 amazing international speakers, Dr. Barbara Taylor and Dr. Jay Barlow, presenting on marine mammal research. So that's probably a little bit different to what we might normally be presenting on in these seminars. It will be a hybrid seminar from the conference room at ARI plus online. So the invite link will be shared shortly. And also just make sure that you've signed up to our seminar mailing list so you can receive the link and recording from this seminar and register for our next seminar, plus sign up for the regular ARI updates as well. So thanks everyone for attending. Thanks to our amazing presenters again for their great work, and hope you have a great day and enjoy the nice weather. See ya.